Namaskar to everyone. Through this series of lectures or discussions, we would be covering cell cycle and cell division. Cell cycle would include everything what is related to a cell from a single to reach to a two daughter cell, and how the cell division occurs. We will discuss all the phases and what all changes that happen in a cell during this stage of division. We are going to discuss those changes and what enzymes or what proteins or what energy things are required during cell divisions. We will discuss all these things during these lecture series. So, first we will try to understand some of the terms before we actually deep dive into the actual understanding of cell division. So, the cells with a single set of chromosomes are haploid cells, and with two sets of chromosomes it is diploid cells. The non-reproductive cells of the body are somatic cells, whereas the cells capable of continuous division are stem cells. Like in bone marrow, human bone marrow, they are in continuous division stage, they are called stem cells, and cells that form gametes or sex cells, they are called as reproductive cells. So, with this basic understanding of what kind of cells are there, we will understand what is cell cycle. So, it is a genetically controlled series of changes that occur in a newly formed cell by which it duplicates its genome, synthesizes other constituents, undergoes growth, and divides to form two daughter cells. It is not just one process, it is a complicated process which require lot of changes into the cell, which are all guided genetically, which are all controlled through your genes. It duplicates its genes or genome, synthesizes other constituents like endoplasmic reticulum, Golgi apparatus, or what we can call it as other cell organelles like mitochondria. So, all these things are synthesized, the cell undergoes any kind of growth, and then finally it divides to form two daughter cells. And all these events occur in a coordinated manner and includes the use of protein cyclins and cyclin dependent protein kinase. So, what cyclin dependent protein kinase is? They phosphorylate proteins using ATP. We all know what ATP is. ATP is adenosine triphosphate, which helps in energy transmission when one goes from ATP to ADP. So, transition from G1 phase to S phase and from G2 phase and M phase is carried out by these CDKs or cyclin dependent protein kinase using the energy stored in the form of ATPs. So, generation time. What is generation time? We hear this term very frequently when we talk about cell cycle or cell division. So, very precisely, generation time is the time interval between two cell cycles, and this may range from a few minutes to few days. When we do a human culture or human cell culture, we can observe that human cells divide once every 24 hours, 10 hours in G1 cycle. We will understand all these G1, S phase, G2 phase and M phase. We will discuss in the upcoming slides what all these cycles or what all these steps are. But for now, we have to understand that there are 24 hours required for a human cell to divide. 1 hour in G1 phase, which is the longest phase, 9 hours in S phase, which is synthetic phase, 4 hours in G2 growth 2 phase, and 1 hour in M phase, that is mitotic phase. For in yeast, from one mother cell to two daughter cell, the division takes 90 minutes, what we call it as a generation time, and in bacteria the generation time is 20 minutes. In every 20 minutes bacterial cells double in number. So, cell cycle or phases or stages or periods, what you want to call it as, has two broad phases. One is I phase and another is M phase. I phase is interphase, which is further subdivided into G1 phase, S phase and G2 phase. So, it is long non-dividing growing phase these lines are really need to be underlined, where we are talking about it is a non-dividing phase, but it is a growing phase. Growth never stops during this phase. The second one is M phase, which is mitotic phase. It is a short dividing phase. Actual cell division happens here, and the preparation of cell division happens in I phase, right. So, what is I phase or interphase or intermitosis? So, series of changes that take place in a newly formed cells and its nucleus before it becomes capable of division again. So, all the initial steps which needs to make the cell prepared for the cell division happens in interphase or I phase, or intermitosis is also named for interphase, because in two different mitosis stages, interphase is the phase that takes place and that makes the cell prepare for the next mitosis. It is a metabolically active phase or energy consuming phase or energy phase, because lot of energy is consumed in this phase. All the basic preparation is done in this phase. So, it is very highly metabolically active phase, and replication of subcellular components including chromosomes happens in interphase and occupies 75 to 95% of total generation time. If we have noted that what is the generation time in human cells, it is around 24 hours, where mitotic phase is 4 hours and 20 hours are being consumed in I phase or interphase. So, about 75 to 95% of total generation time is being used by interphase. So, interphase period of human cells lasts throughout the life for some of the cells, like neurological cells or neurons, because they never reproduce or never divide. So, for them the interphase period is throughout the life. So, the first subphase of interphase is G1 phase, first growth phase or pre-mitotic gap phase, post-mitotic gap phase. So, the cell which has undergone division through mitosis will now further undergo or prepare itself for further mitosis. So, it is post mitotic gap phase. So, mitosis of the previous cycle has completed and now we are entering into another cell division cycle, interphase, and then for the interphase the subphase is G1 phase. So, it is known as post mitotic gap phase. So, the phase between end of M phase of previous cell cycle and initiation of DNA replication. So, it is the longest phase of interphase generation time. If you remember, then the division given to G1 phase is 10 hours out of 24 hours of total time that has been taken by a division, mitotic division. So, nucleus grows in a small extent only during G1 phase, but RNA and proteins are synthesized. Nucleotides, amino acids for histone synthesis, energy rich compounds are formed in this G1 phase or first growth phase. So, G1 phase, when a cell enters a G1 phase, it has three options: either to continue cell cycles and enter the S phase for further division, stop the cell cycle and enter G0 phase for undergoing differentiation phase. G0 is a quiescent phase. We will discuss in detail what is G0 phase, and or a cell once entered in G1 phase get arrested in G1 phase itself, whence it may enter G0 phase or re-enter cell cycle. So, it stays in G1 phase, just the growth phase, and at a later stage when required it may enter G0 phase or re-enter the cell cycle, that is can enter S in the S phase. So, what is S phase? So, S phase is the second phase of interphase. It is called as a synthetic phase due to the replication of chromosomes, or it prepares itself for nucleus duplication. So, DNA molecules functions as templates and form carbon copies of itself. DNA content doubles in this phase from 1C to 2C for a haploid, that is from one chromosome to two chromosome in a haploid cell, whereas in diploid cells it reaches from 2C to 4C. So, the chromosome replication is the main step that happens in the S phase. Duplicate set of genes are formed and new chromatin fibers are also formed, which however remain attached in pairs. They have not separated, that is why the number of chromosomes does not increase, because they have not separated, they have just replicated but remain attached to each other. That is why the number of chromosomes does not increase. They are attached with the centromere, they remain attached, and at the last stage of division they remain, till that stage they remain attached, and at the last stage they actually detach themselves, and then the number of chromosomes change. But at that stage, because it has to be converted to two daughter cells, so again the one daughter cell would have the same number of chromosome as the parent cell. So, you have to remember that number of chromosomes does not increase even though they have duplicated themselves. So, as chromatin fibers are elongated chromosomes, each chromosome comes to have two chromatin threads now, or sister chromatids, which remain attached to a common point called centromere. So, they remain attached. We will see in the coming slides that how they are attached. So, S phase is also called an invisible phase of M phase, because chromosomes prepare themselves for equitable distribution. M phase is mitotic phase where actual cells divide, but in the S phase the most important component of a cell, that is the chromosomes, have divided themselves. So, chromosomes have prepared themselves for the equitable distribution, that is why this phase is called invisible phase of M phase. And that is why the name is given that a synthetic phase, because the most important synthetic component has been duplicated in this phase. Subunits of kinetochores are synthesized in this, and then centromere begins to divide in centriole containing cells to form two centrosomes or centriole pairs. Because the two poles will be formed once the division will start, that is why there is requirement of centriole pairs. So, centriole start dividing and start forming a pair set. In G2 phase, it is the second growth phase or pre-mitotic gap phase. This is the very phase where post this phase the mitosis will actually happen, that is the cell division will actually take place. So, this is the second growth phase. Synthesis of DNA stops because everything has been completed in synthetic phase, that is S phase. Now, formation of RNA and protein continues. It continues from G1 phase to S phase and to G2 phase. They remain in a continuous production phase. RNA and proteins are required for multiplication of cell organelles, spindle formation, cell growth. So, that is why the RNA replication and the protein formation continues throughout the cycle of cell division. So, it prepares the cells to undergo, because you need two sets of cell organelles, you need the spindle formation, and the cell growth is also required, so that they can finally divide into two daughter cell. The new daughter cells need cytoplasm also, nucleoplasm also, different cell organelles also, and during this stage of cell division lot of energy is also required. So, cell organelles are required to create that energy, like mitochondria. Mitochondria is a component where it is a cell factory. So, all the energy has to be formed through mitochondria. So, everything needs to be duplicated for the preparation for the cell to undergo any kind of division. So, that is why these things continue. So, now we will understand what is the difference between G1 and G2 phase. Yes, we have noted that some of the things happen in G2, some of the steps happens in G1, but let us discuss the difference between these two phases, so it makes our understanding more clear. So, G1 phase is the first substage of interphase, whereas G2 is the last stage of interphase, right. The cycle goes like G1 to S and then G2 and then M phase. So, it is the first stage, G1 is the first stage and G2 is the last stage. The available factors determine its fate. We have seen three different fates that are possible once the cell enters into G1 phase. So, entry in G2 or differentiation or continuity of cell cycle. But in G2 there is hardly any choice, because it is the stage when in any case M phase will enter. So, very little choice for cell except to proceed further in cell cycle and go to M phase. In G1 phase, cell organelles do not increase in number, whereas G2 phase finally the cell organelles increase in number. In G1 phase, cell grows in size, but growth of nucleus is very limited, whereas in G2 phase, because this is the final phase before the actual cell division in M phase, both cell and nucleus grow in size. In G1 phase, synthesis of RNAs, proteins and other biomolecules for cell growth and subsequent replication of DNA are synthesized, whereas in G2 phase, synthesis of RNA, proteins and other biomolecules for spindle formation and M phase division are formed. Right, there is a difference that what is the requirement in G1 phase, because it is going to enter into S phase, and in G2 the difference is that it is going to enter into M phase, where we need the spindles and actual transition to M phase, right. So, that is why the RNAs and proteins prepare themselves for DNA replication in G1 phase, whereas in G2 RNAs and proteins prepare themselves for spindle formation and M phase division. So, now we will understand in brief what is G0 phase or quiescent phase. So, it is a stage of inactivation of cell cycle when there cell cycle in terms of cell cycle there is nothing happening which is related to cell division due to non-availability of mitogens and energy rich compounds. Mitogens are the reason that promotes the cell or that makes the cell go ahead with the mitosis. Because there is no mitogens available, that is why the cell enters into G0 phase. Cell remains metabolically active, but not active for cell division. It will not metabolize or it will not make any biomolecules which are required for cell division. So, this is how G0 phase is different from G1 or G2 phase. No growth, no differentiation. The cell functions as reserve cells which can join cell cycle at any time. So, now we will enter into the M phase, mitotic phase or actual cell division phase. Till now what we have studied in interphase through G1S and G2 phase, the cell is preparing itself for cell division, but actual cell division has not happened as of now. In M phase, the cell division will take place. It is the final phase of cell cycle, phase of actual division, stage of separation of already duplicated components, because all the components have been increased in number by G2 phase. So, after M phase, a cell may re-enter fresh cycle of cell division or pass into G0 phase, as we have already seen, and the cell may grow in size and become differentiated. Once the M phase has been completed, there are two options for any cell. It may re-enter fresh cycle of cell division, or it may pass into G0 phase where nothing happens, it stays as such, quiescent stage, it will not do anything, just metabolically active, but else it will not do anything. Or the cell may grow in size and become differentiated. So, see in human body or in any animal or any other plant, there are the tissues which are present, which are the part of cells being differentiated. Tissues are formed once the cells are differentiated. Skin has different kind of differentiated cells, whereas liver has different type of cells. So, these are formed once the cell has been differentiated to do a particular task, specified task. So, a cell may remain as such, or it may finally get differentiated like columnar cell or cell with cilia or villi or or it may be squamous cell. So, they may enter into a stage where they are differentiated to perform different functions. So, after mitotic phase, the cell may enter into that kind of differentiation. So, differentiation phase, most of the newly formed cells do not enter the G1 phase for the next cell division. They grow in size, assume particular shape, come to have a particular function which is called cell differentiation, right? Kidney have different kind of cells, nephrons. Intestine would have a different kind of cells. They perform different kind of functions. Our stomach has different kind of lining. Some of the cells may release mucus. Some of the cells are made for releasing the hormones. Some are endocrine, some are exocrine. Skin have different kind of cells. So all these cells, they grow in size. They don't enter the next phase of division. They just grow in size, assume particular shape, and come to have a particular function. So, what is the significance of cell division? So, before entering into the different phases of M phase, we will just understand very briefly what is the significance, why the cell division is happening. Obviously, cell multiplication is one, and then continuity of any generation or continuity of the body, cell division is required. Like for bacteria, if they don't divide, then continuity of their life ends there and then. Asexual reproduction in lower organisms like bacteria, I just gave you an example. Then growth of multicellular organism. So, imagine if we would have remained the same size as we were born of. Is that possible? No, we need to grow. So growth of multicellular organism is done by cell division. Then differentiation in multicellular organisms. Every cell is not the same in human body or any other multicellular organism. In lion, is every cell same? No. So that is not possible. So differentiation in multicellular organisms required to be done once the cell has been divided. Then maintaining the genetic similarity. Mitosis is a phase which maintains the genetic similarity. Then repair, regeneration. Sexual reproduction is through the reproductive cells that divide and create those cells. Then reshuffling of genetic traits. Meiosis is one which reshuffles the genetic traits, and mutations is one thing where we or any other multicellular organism can adapt to any particular environment.